Hi, my name is Wendy Demas. I represent the Rat Genome Database, RGD, at the Medical College of Wisconsin. RGD joined the Genome Reference Consortium, GRC, to further improvements of the mammalian rat brown Norway version 7.2 genome assembly through the curation of errors brought forward by RefSeq curators in the rat community. Today, I'm going to describe the workflow used to resolve those reported issues. The genome records were annotated with the NCBI eukaryotic genome annotation pipeline. Assembly issues are recorded in the Atlassian Jira ticketing system. The GRC rat issues dashboard pictured here provides public access to individual views of all reported issues and their resolution status. To determine a starting point, RGD provided the summary of issues to members of the International Rat Omics Consortium with a request to identify specific genes of interest in the community. Those genes were used as a filter to prioritize tickets with reported known or potential effects on protein function. The resolution workflow is broken into pre-assessment and resolution stages. A variety of tools provided by NCBI, RGD, and the GRC are utilized to review and resolve those tickets. RG129 is reported to have a 1 nucleotide insertion, which results in frame shift in the encoded CDS and premature termination of the protein. The first step in the pre-assessment stage is to identify the strain listed as the source in the NCBI nucleotide database. In this ticket, a specific strain is not provided. The reported position is reviewed in RGD's variant visualizer tool. These data may indicate that the reported issue is common across strains, as seen here in RG129. The inset box indicates that all reads for this position do not have an insertion regardless of strain. Illumina short read data of the same animal used for reference generation are viewed in JBrowse. These data serve as a comparison to the long read data used to generate the reference. This step allows the curator to compare different reference assemblies and to potentially identify problematic assembly regions. The global view here shows some assembly issues from reference version 6 may have been corrected in reference version 7.2. The long read alignments are viewed in the NCBI Genome Workbench tool. This is the tool where the heavy lifting of the resolution occurs. Alignments, reference version 6.0 and 7.2 are easily compared. In this example, you can see the reported issue occurs in a repetitive region. If an alternative alignment was available, it would be visible below the transcript sequence. However, there are no alternatives to review for this region. Since the transcript sequence is stable, context for assembly versions 6 and 7.2 were blasted against the transcript. This view shows there is a discrepancy in the number of Cs in the region between builds, as well as a mismatch in insertion for reference version 6. Due to the repetitive nature of this region, this is likely an error in the reference. This issue could potentially be resolved with sequence from reference version 6, but is currently stalled. The transcript for OPRM1 was reported to have an insertion in the new genome relative to the old, resulting in a frame shift for ticket RG8. The strain is listed as Sprague-Dalley, which may indicate the reported issue could be specific to the brown Norway strain. The reported position is reviewed in RGD's variant visualizer tool. Brown Norway strain is highlighted in the red box. Other strains also carry the same nucleotide for this location. This may indicate a technical error as all strains agree. JBrowse short read data on the left show the view of reference version 6 has fewer assembly versions as compared to version 7.2. The long read alignment, as viewed in the Genome Workbench on the right, shows an alternative contig is available to potentially resolve the issue. Blast alignments to the chromosomal region show 100% identity between the alternative contig and chromosomal region. The positive slope of the line in the dot matrix view indicates the chromosome and contig are perfectly aligned and in the same orientation. The alternative contig was also blasted against the transcript, and you see the same alignment and orientation. Individual blast shows that the alternative contig and RNOR6 contig agree with the transcript. The alignment of the two contigs for reference version 7.2 show agreement with the exception of the reported error. This supports a region of the alternative contig can be utilized to electronically resolve the reported issue. The TPF file was successfully submitted. The gray ball on the TPF view shows the region of the alternative contig is listed as a contained subsequence. Ticket RG8 was closed and the status is updated on the GRC webpage. RG90 is reported to have numerous indels and mismatches compared to transcript NM012919.3, which causes frame shifts in the encoded protein. The results of the first step of the pre-assessment indicates the reference is also based on the Sprague Valley strain. Variant Visualize provides data for 6 of the 7 issues reported in Ticket RG90. Data show there is both common and unique variants to the brown Norway strain. 4 of the reported issues listed in Ticket RG90 will be resolved using the workflow described for RG8. Preliminary data for this location, the short read alignment is better in the new reference. The error was reported for transcript 012919.3, but other transcripts do not contain the reported missing A and agree with both the primary and alternative alignments. Variant Visualizer shows brown Norway may be unique in this region, and JBrowse review indicates this region is not overly problematic to assemble. Alignment data for both the previous and latest reference assemblies do not agree with multi-ref-seq transcript sequence. These last 2 cases for Ticket RG90 are a good example where additional data, such as other strain genome assemblies, would provide good evidence to address the resolution of those errors. Thank you for your time and attention.